Do you want to be a part of the future? Synthetic embryos and artificial wombs are on the horizon, and they're going to change everything we know about reproduction. In this video, I'm going to talk about how these technologies will impact our society and what we can do to prepare for them. An embryo is the earliest stage of development in a multicellular organism. Typically, embryos are very small and not yet able to live outside their mother's bodies. A stem cell is a type of cell that has the potential to develop into other types of cells in the body. Stem cells can be found in embryos and in adults. Imagine creating an embryo from a stem cell. Now, can you imagine giving birth to a baby without an egg and a sperm while using an artificial womb? A significant new development has given us a better idea of what an answer might look like. In August 2022, two separate research groups demonstrated that they could create sophisticated synthetic mouse embryos out of stem cells rather than eggs and sperm. Even more impressively, they used an artificial womb to keep the embryos alive for 8.5 days, nearly half the typical 20-day gestation period of mice. This was long enough for them to develop a beating heart and the foundations of a brain. This new technology could give scientists insight into developmental processes that have been difficult to observe. There is potential for the technology to be used medically, potentially making it possible to grow new organs for transplants or tackle age-related infertility. What are the scientific and medical potential? And what about the ethical implications of this process? Evolutionary biologists are still only just beginning to understand how organisms evolve from a single fertilized cell to one with hundreds of billions or trillions of different cells. Researchers studying transparent eggs have made significant progress by looking at animals like zebrafish and frogs, according to developmental biologists. Development in mammals, on the other hand, is much more difficult to observe because it takes place within the womb. Even further ethical concerns and tight restrictions make studying human embryos even more difficult. In recent years, there has been progress in generating blastocysts, the precursor to an embryo, from embryonic stem cells, which may offer a useful model for early human development that avoids some of the pitfalls. However, without the womb's nurturing environment, blastoids can only survive and develop so much before they reach their final stages of embryonic development. In August of 2022, a pair of papers published by two separate groups shook the scientific world as they demonstrated the use of an artificial womb to keep mouse embryos derived from embryonic stem cells alive for 8.5 days. The two papers revolved around a machine designed to copy the conditions of the uterus, created by Jacob Hanna from the Weizmann Institute of Science in Israel. This included glass vials that rotated and contained blood serum, as well as a ventilation system that would regulate pressure and oxygen levels. But the artificial womb wasn't the only innovation. In 2018, Magdalena Cernica-Coates and her team discovered that embryonic stem cells grew better when combined with stem cells from two structures key to early development, the placenta and yolk sac. By combining these two breakthroughs, the two teams were able to create synthetic mouse embryos that developed to the stage where the basic body plan starts to emerge. This included complex structures including beating hearts, rudimentary brains, and the beginnings of a gut. The most obvious impact these advanced synthetic embryos could have is helping scientists crack the underlying code of development. This underpins all kinds of critical capabilities such as regeneration, reproduction, and tissue specialization that have significant implications for medicine. The advantage of this synthetic embryo model is that we can genetically engineer the stem cells used to make them. This suggests we can figure out how particular genes contribute to the developmental process. The other group, led by Hanna, has even grander ambitions for the technology. They have created a startup called Renewal Bio that plans to create synthetic embryos from patient cells whose tissues could be harvested for transplantation. While advances in the development of simple tissues like skin and cartilage have been significant, organs with complex 3D structures and vasculature have proven to be more difficult. Hanna thinks that relying on the body's natural growth mechanisms might be a better solution. Among the potential applications the company envisages are collecting blood cells from embryos and transfusing them into patients to rejuvenate their immune system, or using cells from rudimentary ovaries to help women extend their fertility later into life. What about the ethical implications? Efforts to reinvent human biology can raise ethical challenges. While guidelines vary between countries, human embryos are typically not allowed to develop beyond 14 days, which is the point at which they start to develop from an amorphous blob of cells into something resembling a body. One potential solution is to genetically engineer the precursor stem cells so that the embryo never develops a head, avoiding any concerns about experimenting on a potentially conscious being. Although some researchers disagree, the potential benefits for people waiting to receive organs via donation should not be overlooked simply because of concerns about synthetic embryos. But while the ethical challenges around the technology are considerable, experts say technical ones are probably a more pressing concern. The efficiency with which stem cells can be coaxed into embryo-like structures is still very low, with less than 1% of all cell cultures doing so successfully. And even when they do, the synthetic embryos feature many defects that raise questions about whether they are truly useful models for development. So, why should we care? We are probably still a long way from this technology making it possible to combat aging or regenerate organs on demand, but it's important not to understate its potential. Despite their cautious assessment of the road ahead, experts liken this development to the birth of Dolly the sheep in 1997, the first mammal to be cloned. While there is still a long way to go, these capabilities do open the door to a new era of regenerative medicine that could have major implications for human health and longevity. And with plans to commercialize the technology already underway, significant developments may come sooner than many think. Leave your comment about how this will impact the future of humanity. Thanks for watching.